For DNA to replicate, the double helix must unwind so the DNA polymerase can use the separated strands as templates to synthesize new double-stranded DNA. And for the replication to happen, there's an origin of replication where the unwinding begins with the help of the initiator protein. And an enzyme called helicase opens up the strand. So this zipper right here is going to symbolize helicase. So it opens up the strands so DNA can be synthesized. And helicase, it breaks down the hydrogen bond at the replication fork. And as you can see by my demonstration, it unwinds the double helix. As the double helix unwinds, the single-stranded binding proteins bind the single strands to keep them separated and stable while it's opening up. And as the DNA unwinds, the enzyme called a gyrase in prokaryotes kind of helps reduce supercoiling and torsional stress on the strands that are being separated. As you can see, I have labeled this 5', 3', 5', and then 3', because DNA strands runs in opposite and anti-parallel direction. Since DNA replicates in the 5 to 3' direction, this indication can help us recognize which is our leading strand and which is our lagging strand. So if we were to replicate both strands, this strand will go from 5' to 3', so we know this is going to be our leading strand, and the bottom strand goes from 3' to 5' out, and as it does that, there's going to be like little fragmented DNA, so that's how you know this is our lagging strand. And like I said, it's going to have fragments, so our lagging strand replicates discontinuously, and our leading strand replicates continuously. And the nucleotides in the DNA, like the DNA that we're opening up and unwinding, are bonded by the phosphodiester bond, which are synthesized by the RNA polymerase. To begin the replication of new strand, the RNA primase synthesizes these this RNA primers to allow DNA polymerase 3 to bind to it. So show what DNA polymerase 3 will look like. So it would bind to this primer, and then it would read the code. And since this is a lagging strand, it stops, and then there's another primer right here, and then it reads from that, binds to that, and then it reads down. And as the replication forks opens up, new primers are placed on the lagging strand to replicate the DNA. But for the leading strand, since I said it replicates continuously, one primer is just placed at the 5' end, and then the DNA polymerase 3 binds to it and just reads it continuously as the replication fork opens up. This discontinuous replication is what prompts little fragments of replicated DNA to be on the lagging strand. And these fragments, these little fragmented DNA that are made, are called Okazaki fragments. And as you can see, these fragments aren't continuous, like I said. So to kind of like combine the Okazaki fragments to make one continuous strand, DNA polymerase 1 kind of like replaces the RNA nucleotides with DNA nucleotides to kind of like close up the gap. And it does this for all the RNA primers that are on the replication fork. So it replaces them with DNA nucleotides instead of RNA nucleotides. So there's still like a little space here that needs to be connected. So the ligase, which is what kind of like binds it all together at the end, the ligase kind of comes in and like close up the gaps and connect them.